Welcome, everyone. We are live for our innovation webinar series. I am Madeline Berg with seedworld Group, and today we are going to dive into one of the most powerful tools in modern plant breeding, which, of course, is genomic selection. And genomic selection holds huge promise for plant breeders: faster genetic gain, more confident decisions, streamlined workflows. But turning that theory into practice is where a whole lot of programs get stuck. And that is where we are going to be talking today. I am looking forward to a very useful, practical, hands-on discussion about how genomic selection can be applied successfully in real-world breeding programs. So we have 3 expert panelists with us today. Before each speaks, I'm going to tell you a little bit more about that individual. But first, let me just start off by introducing all 3. This is truly a spectacular list of people with incredible insight and knowledge. So I will just put in a plug now to say, if you've got questions throughout this presentation, and I'm guessing that you will, feel free to add your questions in the chat. You'll see that on the right-hand side of your screen. And we will get to as many questions at the tail end of the presentation as we can. There's a lot to cover. So we will do our very best to reserve a good amount of time at the end of the presentation. So the 3 that we have before you today: Dr. Enid Perez-Lara is a plant breeding expert with agronomic software. She is going to walk us through an overview of where we're at, why it matters so much, particularly some of the challenges and how those challenges can be overcome. Our second speaker is Dr. Diego Harquini, who is the Assistant Professor, Integration and Application of AI and Omics in Plant Breeding at the University of Florida. For anyone who wasn't totally sure, just to clarify, omics is the large-scale biological data discipline that ended omics. So genomics, of course, but phenomics, transcriptomics, proteomics, all of those. And our third speaker is Dr. Caio Canella Vieira, who is the Assistant Professor of Soybean Breeding at the University of Arkansas. And he is actively using genomic selection technologies and can speak to some of what has worked in this lab. So our goal today, as I said, is a fast-paced deep dive into genomic selection. We're going to cover fundamentals to benefits to challenges and solutions. And as you'll notice at the bottom of this slide, the webinar is being recorded and it will be available after this live webinar. So it will be sent out as an on-demand link to all registrants of this webinar, and it will also be posted to the seedworld website. So we are going to start today's discussion with Enid. Let me just tell you a little bit more about Enid. Enid is a seasoned plant breeder. She has been in the business for more than 20 years, 24, I think. We'll have her jump on in just a moment and she can clarify for me. She is a passionate advocate for diversity in agricultural sciences. She has worked across various crops, supporting research, innovation, and technology adoption on a global scale. And she is currently serving as the Industry Engagement Manager at Agronomic Software, where she collaborates with breeding programs from right around the world to integrate cutting-edge digital tools that enhance decision-making and efficiency. Beyond her technical expertise, she is a huge champion of women in plant breeding, actively promoting career opportunities for young scientists and fostering international knowledge exchange to drive innovation in the field. Enid, we're delighted to have you. If you can just come on camera here, there you are. I am going to jump off camera and I'm going to pass the control of the slides over to you. We're excited to hear what you've got to say. Go ahead.
Thank you so much, Madeleine. And thank you, SeedWorld, for the wonderful organization of this webinar. Thank you, Diego and Caio, for being here today as well. Hello, and hello, everyone who is present in the room right now. I want you to imagine this. You've just made your selection for the season, and instead of waiting for a full year of results, you already know which plants will perform best. This is the magic of genomic selection. So your cycle has cut in half, improve accuracy, and you are years ahead of your competitors, which is very important. So the reality is that that's how in theory it works, but many programs still struggle to make it work. So as Madeleine said, I am almost 25 years now in plant breeding, working in plant breeding across multiple crops and continents. And I've seen firsthand what is possible with genomic selection and what is actually being used. But I have been a witness as well of the gap that exists already in many plant breeding programs that cannot get it to work. So genomic selection is not just a breeding strategy. It's not just a tool that you use randomly. It's a way to respond to urgent challenges that we are facing right now, like weather changing, etc. So it allowed to faster cycles, meaning that we release varieties in less time, and we are able to respond to new pests, for example, or changing weather patterns. Everybody knows that this is a huge problem. So it also involves or helps to get higher accuracy that ensures that we use our limited resources. More plant breeding programs have limited resources in a more effective way, and also allows to get better or greater genetic gain. And this allowed to get resilient and non-productive varieties that will go ahead and help farmers and consumers in general. But what happens if we don't move forward? So the cost of not adopting genomic selection is the opposite of this. Every year we delay the adoption, we lose valuable time. The breeding cycles remain longer. This means a slower adaptation to extreme weather or new diseases or simply shifting market demands. And it also means that we miss opportunities. Varieties that could be helping farmers are left undeveloped. And in plant breeding, everybody knows that any delay is a risk. So if the cost of doing nothing is high, what is holding so many programs back? And the issue is that there are a lot of challenges to adoption. Across many breeding programs, the barriers are strikingly consistent. So genotyping and phenotyping large populations remain very, very expensive. Infrastructure like labs, field station, rentals, computing power are also limited. The data lives in scattered, non-standardized files, making integration slow and prone to errors. Teams often lack of trained specialists like bioinformatician, data scientists, or quantitative geneticists. And even when the resources are available, the return on investment is very uncertain because when you release a new variety, there is not any guarantees that you will have the return as soon as possible. And also there is a big resistance. Changing from something traditional to something new is never easy. So among all these challenges, there is one that always raises to the top, and it's the cost of genotyping. The cost of genotypings vary widely. So we can find genotype by sequencing, for example, ranging from $10 to $25. This was from 2017 when I last time did it. Target resequencing can be $18 per sample. Whole genome sequencing is in the hundreds, so 100 to 500 per sample. SNP arrays are very comprehensive, but they demand an investment up front. So even the automated DNA extraction can be complicated. And this is doesn't matter if it's few dollars or hundreds. The problem is like when you escalate this to thousands of lines in breeding programs, the cost adds up and becomes very, very expensive. So why the cost of genotyping becomes so expensive? There is several aspects of it. So we want more data to apply to genomic selection, but more markers involve more cost. Some genotyping methods are more cost effective, other ones are not that so much. And there is also a difference in the place in the world where these service providers are located. For example, there is not the same price in China than in the United States or Europe. I remember I sent my samples to China because it was cheaper at the time. But also the throughput or the technology used can make a difference in the price. The good news is like breeders are already finding the way to reduce the cost of this genotyping. So, for example, they combine methods, make it more practical. So they combine amplicon sequencing with genotyping by sequencing. They also use targeting resequencing, just using some genome regions. And they do multiplexing. So they barcode their samples and put them together to reduce cost. Or even if they are using, for example, Illumina technology, they will go ahead and prepare the libraries in-house to reduce the cost. But genotyping alone doesn't solve the challenge. There are other costs to consider. Actually, one of the highest costs is phenotyping. And this is because we need to plant large extensions of land to be able to phenotype that material several times. And then not just that, we need manpower to take data, and it needs to be accurate. And that's one of the higher costs. Also, creating and maintaining reference populations is very, very expensive. And also data silos. So we collect data generally using different platforms, using different files, etc. And that becomes complicated. The infrastructure, computing, storage, software licenses, etc., make it expensive. And also a specialized expertise. As everybody knows, statisticians and bioinformaticians are in short demand nowadays, and they are very expensive as well. They are in short supply. And also there is a gap in the decision making. So the prediction doesn't translate directly to breeding. So how do we make genomic selection practical for everyday breeding? So we have surveyed the breeders we know, and all of them have specific requests. For example, they want centralized connected data, user-friendly platforms that reduce the use of those specialists that are in short demand. They want prediction tools linked directly to breeding decisions, and they want action-oriented workflows where analysis drives the advancement. And of course, they want that the infrastructure is scalable to the program size. So if they have bigger program, they want to have space for it. And if they have a smaller one, they want, of course, to pay less. In short, they want a genomic selection is a practical tool and not an experimental tool. This is where our role and integration becomes a game changer. So how we work out this demand? So we have integrate in everything into the same process. So genotypic and phenotypic data lives in the same environment. The predicted model, whether G-block, Bayesian, or custom, the details that Caio and Diego will talk about are built in. The data can be imported directly from sequencing providers. So this is fed in directly into the system, and the predictions can feed straight into the crossing or advancement list, helping the breeder to make their decisions. And this way, genomic selection stops being a side project and becomes a part of the breeder's workflow directly. And this is how we see our role in this community of breeders. At Agronomics, our mission is to help to feed the world. So our responsibility is not simply to sell tools to breeders, is to make, for example, genomic selection accessible for all breeders. And by removing friction, we make adoption easier. And the faster genomic selection is adopted by all the breeders in the world, the sooner we can respond to climate change, to food shortage, to nutritional needs, or to market demands. And integration for us is a way to give back to the breeding community as well. And the impact is already visible. We have examples here. For example, we know wheat breeders that have reduced their cycle for a year. Also, vegetable breeders who have identified winners before planting, saving land, time, money, etc. And national programs that are already competing globally with multinational. So what does this mean for us and for the breeding community? This means that genomic selection is not just a tool or an innovation randomly selected to be different or to be in the last fashion. It's an accelerator of progress. These barriers that I mentioned are real barriers, and we can help to solve some of them. So our role is to remove some of these barriers for breeders so they can actually focus on what really matters, that is delivering resilient varieties and help to feed the world by 2050. And at the end of the day, all of us, the breeders, we at Agronomics, everybody here in this room, we are consumers as well. So this is important for us. And I would like to take this conversation to another level. So I'm dropping here a QR code so you guys can scan it and you can contact me directly or to my team to keep the conversation going, share your thoughts, explore how we can make genomic selection practical for your program. And I would like to thank everybody again for the attention. And I want to remind you that genomic selection is just a tool. And it's a very important tool nowadays in the breeder toolbox, but the breeder is still at the driver's seat. Thank you very much.
Thank you, Enid. I'll just leave this on screen for another moment so that people can collect that QR code. As I mentioned earlier, we are going to be sending out a on-demand link, so you can also have access to Enid's email through that and this QR code. Our second speaker is Dr. Diego Jarquin. And Diego, I am especially appreciative that he has joined us today because Diego is not feeling his very best right now. He's got quite a bug that he is fighting. So we will all be very understanding of the fact that he might have a Kleenex beside his nose. Diego, if you want to jump on the screen here with us, I will just introduce you. You are the Assistant Professor of Integration and Application of AI and Omics in Plant Breeding at the University of Florida. And let me just tell our audience a little more about you. You received your PhD in statistics from the University of Postgraduate Education in Mexico back about a dozen-ish years ago, 2012. And then you went on to postdoc training at the University of Alabama, Birmingham, and the University of Nebraska-Lincoln. And later you were promoted to research assistant professor and research associate professor. Diego is a statistician who merges that concept of statistical methodology with AI, computer algorithm development, data science, and collaborative work with plant scientists. So really pulling all those pieces together. You have a very active research agenda on advancing how prediction models are developed for selection purposes in plant breeding. And as part of your work, you have an extensive library of peer-reviewed publications. Am I right in saying 90 manuscripts, 4 book chapters for about 5,600 citations? So you've been quite prolific. And you also have to your name several very notable awards. You received the Early Career Scientist Award from the National Association of Plant Breeders, and recently received, I think it was last 2024, the UF IFAS Plant Breeding Innovation Award, and the 2025 UF Excellence Award for assistant professors. So big congratulations are due to you on that. Can I turn the floor over to you, Diego?
Okay, yes, thank you. Hello, everybody. First of all, thank you, seedworld, for the invite, and thank you, Enid, for involving me in this great project. Today, I'm going to talk about what's new in genomic selection and the main considerations for successful implementation. So this is the outline of the short presentation. First, I'm going to talk very quickly about genomic selection. It's great, Enid already introduced the topic, so I'm going to use less time for that. And then I'm going to talk about beyond genomic data. So what is going after we are able to collect more data? How can we use that information? And also some tools for integrated multi-omic data, what can be done. Enid already presented some ideas of some products that can be used. And also we developed some free tools that people can access. And finally, I'm going to talk very quickly about some new modeling strategies that we have been working the last months. So first, I'm going to talk about genomic selection. As you know, many of these economical traits are complex because they are affected by a very large number of genes with a small effect. But more important, these gene effects are modulated by environmental conditions that these genotypes face in fields. In a way, the genotypes and environmental factors interact in many complex ways. So this is the concept of genomic selection in a nutshell. As we know, genomic selection allows to increase the testing capacity of the breeding programs by far. And the concept is very easy to explain. So initially we need a data set with phenotypic and genomic data, and then we calibrate models, and it could be any model, very elaborated AI method, or use the GBlood model. And then we can conduct prediction on test candidates. And then in theory, we will provide these results to the breeders, and they can use those for help the selection process. As I said before, seems like very easy concept to understand, right? Initial data set with training for training the model. Why? Because phenotyping costs are increasing, and why not? We already have some data. We can use that for conduct the predictions, and then from there use those results for selections. However, things are complicated because, as we know, the breeder's labor is to develop genotypes that perform other elite varieties in a wide range of environmental conditions. In a way, they have to conduct multi-environmental trials in order to select the best genotypes. And many times it happens something like this. The performance of the genotype is going to vary from one environmental condition to another. In this simple representation with 3 genotypes, we see that there is not a genotype that is going to outperform the others, right? One is constant, the other is good in one place, but in another, and the opposite for the other one. Using my breeders have to deal with not just 3 genotypes, with thousands of genotypes in many different locations. So in this case, there are 3 different options to pursue. We can ignore the fact that we have genotype by manual interaction, and we can just select those genotypes based on the means, but that won't ensure that we will have something better than the elite varieties. We can reduce it by selecting genotypes within location, something like local adaptation, but as we know, weather conditions change from one environment to another. And finally, we can exploit, and the way to do it is by using this information, this being collected in multiple environments, to reduce the phenotyping costs and to increase the testing capacity. So let's talk about what's happening beyond genomic data. So this very nice figure that was put together by Julian Garcia and Moises Nascimento shows what we call the omics verse, and it sounds like a superhero movie where we have many different sources of variability playing at the same time to try to explain trait variability. So besides genomic information, we have transcriptomic, metabolomics, phenomics, that is being collected with UAVs, proximal and remote sensing, environmics, where we can have access to pretty much any weather information in the world by just knowing the coordinates, and other omics like ionomics, we can have information about the concentration of the elements in the tissue, epigenomics, etc., etc. So now the question is, how can we make efficient use of all this information that is now available? And then it has to do a little bit with the model. As I said before, genomic selection. One of the advantages of working with this methodology is that we can conduct selections by skipping the planting day. And it sounds very interesting. Just imagine if you have a very large data set of genotypes, let's say 100,000, and then you are dedicated to select the top 1%, you can either go through a series of selections year by year, or you can try to identify what will be the most successful genotypes in very few evaluation cycles. Some of the disadvantages is that it depends on trait heritability. Sometimes we might have some genotypes with very low performance. The second strategy is phenomics, where the advantage we have seen based on the experience is that usually we have higher accuracy in genomic selection, and combined with molecular markers, the results are slightly better, right? The disadvantage obviously requires to observe genotypes in fields, so we have to plan those. We have to develop a sophisticated hydrophenotyping platforms, and sometimes it requires some continuous monitoring. One of the other problems is that these covariates not only capture genetic signals, but also environmental pulse that also might include environmental factors and interaction with genes. So sometimes these signals are not transferred to other environments in a sort of phenomic markers, so we can reduce predictability predicting new environments. Then environmics, the advantage is, as I said before, it's easy to collect data from public databases. We just have to know the coordinates and planting date, and it's help to characterize differences between environments. Disadvantage is the set of available covariates might not be enough to fully characterize differences between environments, and we won't be able to borrow the information as we expect. So also we have some modeling challenges. So prediction framework. So we have the statistical model is just an oversimplistic representation of the reality. We are expecting that every single component in the model is going to explain some portion of the variability. So in this case, we have to explicitly declare the variables in the model. On the other hand, we have non-parametric AI models that work in a sort of black box type, and these are very easy to implement. These capture nonlinear relationships, and this is good. Usually they help to improve predictability. However, dealing with multi-omic integration, sometimes the machine could trick the user by just focusing on variables or layers to minimize a loss function. As we know, breeders care about genetic gain. I mean, it might be the case that other sort of variability, right, is gonna explain most the variability of the genotypes in fields. Other alternatives are the crowd models that relies on some specific constants. These constants provide valuable information regarding the development of the genotypes, the growing stages, yield components. However, it might require extensive studies to find these constants to be able to extrapolate a wide range of environmental conditions. And finally, the drawback dealing with multi-omic integration is that adding all the data in a bulk fashion, as we know, can help to improve predictability. However, it's going to be very difficult to create orthogonal set of information. Why it is important? Because we really want to know what the specific proportion of the variability that's been explained by each one of the single components. In the absence of one of these variables, the remaining variables will fight to explain that variability that is left. So in that case, it's difficult to truly quantify the contribution of a specific variable or set of variables. It's not like going to the supermarket. We pay we get the receipt, we know how much each one of the different items cost. In this case, we wouldn't know. And the next question is, we are dealing with some sort of missing heritability, or we say that whatever we're not able to explain in the field has to do with the heritability is missing. But something that we have been working lately is something that we call missing comparability, that sometimes it might be the case that we want to compare the performance of the model that we have with something that we already observed with the phenotype. And it might be the case that that's not the best either. So, for example, dealing with special adjustments, we have the better we the better we are trying to untangle this field variability, the worse the worse or the less the breeding value is gonna look like the phenotype in field. So if we're able to isolate the genetic component in a good way, so probably that genetic component, it has to look less than the phenotype, and then we can select based on the phenotypes. In this plot, we have an example of 4 different sources of variability. In the inner square, we have the real breeding values. In the outer square, in the top, we have the correspondence with the phenotypes, and we have there a correlation of 0.65. So we have a lot of crossing points. In the bottom, we have the correspondence with the field variability, and there is no reason to have genotypes aligned with some specific pattern in the field. So the correlation is 0. In the left side, we have the correlation between, or the correspondence between the breeding value and the adjusted value within each one of the cells in the field. And in the left side, the right side, we have graded the correspondence between the breeding value and the predicted breeding value. As we can see here, actually the models are not that bad. The models are bad or look bad when we are comparing with something that's coming directly from the phenotype, but when we compare with the real breeding value, obviously in the real life we won't never know, the results are way better. So now I'm going to talk very quickly about some tools for integrated multi-omic data. And one of the products that we have in the lab is Chido. Chido is a no-code application that aims to remove technical and financial barriers associated with modern GMI interactions in a prediction context. It stands for Characterization and Integration of Driven Omics. And this was initially developed by Francisco Gonzalez, who is now a PhD student in Oregon, and is modern apps lead at Google North America. In collaboration with Julian Garcia, ex-Inter at Google, and later with Victor Segues, now breeder at Bayer, we put together this great implementation. And this is how the implementation can be accessed. So we can access on demand, right? These are the interface. You can upload your data. You can create the models, train, validate the models, and etc. So this is free. Also, you can download in your own computer, and the capacity is going to depend on the capacity of your computer. And finally, some new modeling strategies. So as we know, we can now test many different models to see whether we can improve predictability or not. But a new fashion of the models is to combine all of them in something that we call stacking ensemble. And this work was developed by Moisés Nascimento and Ana Carolina from University of Viçosa in Brazil. And the idea was to take the best or the more appealing properties of the different methods. So G-block model, the quantile regression, Mars, quantile random forest. So each one of these base learning has something positive. And the way how to associate them can help us to have a better predictor. In this case, we tested this using a coffee data set for 4 different traits, and we found that for yield, predictability was improved almost 90%, total number of fruits, 38%, leaf mineral infestation, almost 200%, and circuit porosis, 15%. So overall, it's clear that when we combine the outputs of the different base learners in an informed way, we can improve predictability. And finally, something that has been done lately by Victor Segues was to take advantage of the GPU acceleration for parametric models. So many of the AI methods were developed, or implementations for AI methods were developed, or created some these improved libraries to speed up the process. So we have seen not significant improvements in predictability by using AI methods. So we are trying now to use those libraries to improve, right, the ways how we can run the conventional methods. So he used Rapid's ecosystem, QDF, and CuPy libraries together with Hypergator, the supercomputer that we have here at University of Florida. And he tested the new implementation using a data set of 2,500 soybean genotypes evaluated in 340 environments, or genotypes in all environments, for a total of 850,000 data points. He used an HPC where he took advantage of one NVIDIA A100 GPU. And these are the benchmarks. So we wanted to know how long does it take to run a conventional model, either with manufacturers or with interactions. And at some point, we found that actually we doubled the size of the data set. We are not even doubling the running time. The running time increased just by 1.7. We started with 100,000 data points. It took something between 5 to 7 minutes to run until 850,000. That took like 40 to 51 minutes. And that was all. So, sorry, if you want to learn more about this, every year we are running a short course here at University of Florida. So next year we already line up the multi-domain integration for AI genomic prediction breeding with the different approaches. Save the date. If you don't have anything to do next year around the summer, July 6 to 10, you are welcome to attend to our short course here in Gainesville, Florida. So we have instructors from multiple universities, research centers, and private sector. So with this, thank you. I'm going to turn to Dr. Kai.
Thank you so much, Diego. We very much appreciate you being here, especially when you are not feeling your very best. Our next speaker is Dr. Caio Canella Vieira, and he is the assistant professor of soybean breeding at the University of Arkansas. Caio leads the university's soybean breeding program. Before he joined the University of Arkansas, he earned his master's and PhD in plant breeding in genetics and genomics from the University of Missouri. His program in Arkansas targets the development and release of high-yielding conventional and herbicide-resistant soybean cultivars with broad biotic and abiotic stressor tolerance, as well as improved seed composition. The program puts efforts into identifying and incorporating ecologically important traits from genetically diverse accessions into modern, high-yielding genetic backgrounds. Substantial efforts are also made to maximize the efficiency of the soybean breeding program through the implementation of genomic prediction strategies across various stages of the pipeline. Caio, thank you very much for joining us. I will pass the floor to you.
All right. Thank you very much. Thank you, Enid, for putting this together. Thank you, Diego, for joining and inviting me to participate as well. It's a pleasure to be here today. So there's a lot of people in the chat and a lot of conversation going on. So it's a pretty exciting venue. So today we have limited amount of time. So my goal is really to go over some of the major things we are doing in our program using genomic prediction, and maybe open for discussion on different things that can be done there. All right, so overview of our program. So we are a public breeding program here at the University of Arkansas. So really the goal, the reason why we exist is to supply growers here in the state with varieties that have the traits that we're looking for, right? So we are a program that does the crossing, selections, and testing locally, which obviously you're going to be targeting a lot of the traits that the farmers are looking for. When you look at priorities of the program, we like to joke that our first priority is yield, second is yield, and the third, if you guess, it's also yield. I heard that many times from both grad school advisors, Dr. Holman Chen and Dr. Grover Shannon, which were one of the most legendary soybean breeders here in the Southern United States. It's a pleasure to be trained by them. So our program is mostly done on a conventional background. So a lot of initial crosses and the diversity generation are done on a conventional background. And we use a mix of foundational breeding, which is tried and proven, right? A lot of the basic breeding works very well. But we also try to use new technologies to make the process more efficient. And I think that's if there's one home take message here, is that a lot of this are tools, right? It's not necessarily changing what we know about breeding, but are tools that can make the process more efficient. So besides conventional, we also develop varieties with both the major herbicide traits, both the enlist and the extend flex. So we'll touch base on that in a second. And as previously introduced, and I appreciate the introduction, a lot of the focus that we do is obviously the high yielding materials, but also resilience to biotic and abiotic stressors. And really the reason with that is so we can maximize adaptability and stability within our target environment, which is Arkansas and the Mid-South. And I want to include both the logos from the commodity boards, who are really responsible for a lot of the funding for the work that we do. All right, so overview of 2025. Past 2 years, we have released 3 maturity group 4 varieties. It's been a big shift from maturity 5 to 4s here in Arkansas, so it's nice to have those materials coming out. And we're also having the first generation of enlist materials coming out of the program being released this year, and then going through seed production and all of that. But I want to include this slide to give you a little bit of perspective of the magnitude of testing, right? So one of the things that we're really pushing to do here is to get broader exposure to different environments in the Mid-South in general. So most of our stages of the breeding program are going to be across not only Arkansas, but different states. But when we get to the regional, which would be the latest stages, we're looking at almost, over 30 locations per year. So we're trying to increase the number of locations to be competitive, right? To be able to make better selections instead of relying in just a handful of locations. So moving more towards the purpose of this presentation, I want to include what is a typical, traditional breeding pipeline. Obviously, people may do this in a million different ways, but this is back of the paper recipe of some of the typical breeding pipelines. I imagine a lot of you in the chat are familiar with breeding, so everything starts with crossing. A traditional program, we try to fix or implement favorable alleles, mostly for major traits, either being disease resistance or abiotic tolerance. We use off-season nursery to go from the F1 seed into F4 or F5, sometimes earlier, but usually F4 derived. That opens up for progeny rows. This is the stage that we're going to increase the amount of seeds, make visual selections to discard anything that has no potential. And from there, things move into yield trials, right? And that's when things really get extensive in terms of resource allocation and different things. When you go through this, you can go, basically complete the whole breeding cycle within 6 to 7 years. It works, right? We know it works. It's a lot of the things that have been done over the years, but it can be more efficient. There's ways to make this process more efficient. And to give you a perspective on the numbers, if you're reading on the slide, you may be able to see that things start with about 25,000 progeny rows, which would be the first stage. And we're cutting down 90 to 90% per year. So you go from 20,000 to 1,500 to 150, all the way to identifying the very best 2 or 3 varieties. So now with genomic prediction, and Enid and Diego did a great job introducing the topics. I'm really not going to cover too much about this. There are ways of rethinking the breeding pipeline using this tool to make things more efficient. So one of the things that we're trying to move towards here is, first of all, how we're designing crosses, right? This is one of the most critical steps, and I think everybody would agree with that, in a breeding pipeline, because if you make the wrong crosses, there's no magic to generate good things, right? So that's really the foundation. And then the other part that we're really trying to manipulate with genomic prediction is this whole early testing in the program, right? So early testing, we have too many materials, too little seeds, and limited locations that those can be allocated. So how can we properly and accurately identify which one of those early genotypes should be tested in large scale and which one should be discarded? So going through this, we can reduce the cycle, right? The length of the cycle. We believe we can identify things with a little bit more precision than just relying purely on visual selections. And with that, if you consider, what the genetic gain equation looks like, we can manipulate a lot of those variables in a way that we can maximize genetic gain. So not to run too much of our time, I want to just show a few examples of how we've been using this. So first of all, in terms of crossing block, this is a very costly process for a lot of breeding programs, especially when we're looking at development and generation advancement. If you have 100 parents in your crossing block, you can get almost 5,000 different crosses. And again, it costs money. So looking at previous data, both from Arkansas and from Missouri, which where I was during grad school, we can see that a lot of populations were developed. Arkansas about 1,200, Missouri about 1,800 populations. But the number of populations that end up working, right, the ones that end up going to the regional trial and deserving to have more testing, were very limited. All right, so in terms of Arkansas, we're looking at about 50% of the lines that made to regional, they came from only 10 biparental populations. And in Missouri, about 50% came from only 29. So we're developing thousands of populations going through all that work for only a handful of those to really work. So there's got to be something regulating the reason why some of those work and some doesn't. So what we tried to do here, we started with about, we've been genotyping close to 1,500 materials every year now. And what we did is we simulated all possible combinations between all the, preliminary and advanced materials in terms of potential crosses. And what we tried to see is, okay, now if we have the genomic data and the simulated progenies, now we can predict how those progenies would potentially perform and also identify which one of the parents could be the best combiners. We know in maize, there's a widely used concept of general combining ability. So we don't necessarily talk too much about that, but there's maybe potential in that direction. So the first thing we saw is that the general combining ability of genotypes, so how well they are as parents, is related to yield as we would expect, but it doesn't explain it all. So I don't really have a pointer here to show you, but if you're looking towards the right of this figure, there's a lot of high yielding materials that may not necessarily produce high yielding progenies. And we see a lot of that, right? I imagine there's a lot of soybean breeders in the chat. There's a lot of crosses we expect are going to work. The parents are great, but the progeny is not. And vice versa. There's a lot of crosses that the parents may not be so great, but the progeny work out. So with this, we can get a better idea of who are the best combiners. And the other function of this whole approach that we're using is how can we do an early recycling of materials, right? Think about breeding cycle. The sooner we can complete the cycle, the more we can generate better materials. So the one way we're using this is we go through materials that may not necessarily have been through too many testing yet and start crossing with them earlier based on the predicted general combinability from each one of them, which is nice. So we can get things in the crossing block much earlier than we would before. And the other thing we can do, which is quite interesting, is that whenever we bring a new genetic diversity into the program, we don't necessarily need to do that in the dark, right? Guessing how they would perform. We can also start predicting potentially how well that material would combine with our germplasm. And lastly, but not least, it's also really cool. So I've been in this position close to 3 years now. So there's a lot of materials developed that I have no clue what they do, but now we genotype them and we can see that there are some pretty old varieties from 1996 that have the potential of being good parents. So there's a nice way of rescuing genetic diversity that may have been lost throughout the years. And lastly, move forward here on the early testing. This is, I think, is one of the challenges, part of the breeding, because of the problem. We have very little seeds, too many materials. Obviously, you cannot test it all everywhere. But what I want to touch base here is in terms of logistics. We talk a lot about different models and different applications, but logistics seems to be one of the major constraints, at least for us. I would imagine in a lot of public breeding programs, because the investment to get genomic data is too high, right? So if we're going to go through this, we got to have something in return. We got to be able to generate some sort of gain in accuracy and whatever that justifies the investment. So one thing that I want to touch base, if you've been following some of what I talked about, we make crosses in the summer. If you see there, Y0, and we get those F5 derived seeds in the spring 2 years later, right, using off-season nursery. So basically where we have highlighted in red is when we would be able to get genotypic data and make predictions. But in a situation like this, we would go through the investment, hundreds of thousands of dollars if you put this in a private scale, millions of dollars. But by spring year 2, you wouldn't have enough seeds to really maximize the decisions you're making. At the end of the day, you would still have 100 seeds in hand. So you go through all this process, but you're not really necessarily decreasing the length of your cycle or increasing your testing capacity. So one of the things that we had to think about here is that we got to this situation, right? We're making predictions, but what's the gain? What are we benefiting from this? So now we're slowly shifting towards indoor crosses, so moving away from field crosses in a way that we can do, instead of relying on summer crosses, we're doing this sometime during the spring. Why? Because we still have the same window for genotyping, but that allows us to do one extra cycle to have enough seeds for testing. So in this way, we can maximize really the benefits of this tool, which is getting predictions, knowing which one are the superior materials, but still having seeds to be able to test them in the field. So showing some real data, this is work we did with Diego back in my grad school, where we're trying to see, can we identify some sort of covariates that are somewhat fixed over the years, right? We talk about environment. Environment's pretty tough to predict. And soil texture in this context seems to be something that was pretty consistent over the years. So are we able to understand maybe perhaps the interaction of some genotypes with the soil, and is that going to provide us better accuracy in terms of prediction? So what we've seen here, in a matter of time, I'm going to skip some of those things, but we saw that adding the soil texture as a covariate helps us to somewhat structure the error term, yielding better prediction accuracies. The other thing I want to touch base really quick, we try to look at this a little more towards a classification problem, so not necessarily just a regression, but how well is the model capturing the best materials, the top predictors, or vice versa? How well is the model capturing the lowest performing materials? So in a cross-validation too, which is, given is that one of the easiest scenarios for performance, we see that if you're using this model, genomic prediction in general, and you're selecting things that were predicted to be the top 50%, you're going to be, 96% of the time selecting something that was above 50%. So your accuracy, your practical accuracy is pretty decent. And then we did something similar. So we looked at, if you're following those different stages, preliminary, finals, regionals, and so on, we look at the CV00, which is really new genotypes in new environments. And granted, your accuracy comes down, but if you may not be able to see on the coloring, you don't really discard anything that was potentially a release based on the coloring here. So if you're using this model and you're still trying to discard, let's say 50% of the lines, you're not discarding anything that made to be a release. You may discard something that got to regionals or something, but the very best product was not discarded. So even though correlation accuracy is low, your practical accuracy is pretty decent. And lastly, to finish, this is work we're conducting now from my master student, Rafael, and following up on this idea of classification. Can we really be able to simply classify materials into high yielding or low yielding, especially in the early stages? I saw some of the questions before there. I personally don't think you're going to be able to predict what is the best genotype, but you may be able to predict what's going to be the worst group or maybe the best group, right? I think the very best one will still be based on phenotypic trials and extensive multi-environment trials, but perhaps we can reduce a little bit the magnitude of testing. So that's what we try to do. We classify materials into high and low based on the performance of the checks, and we use classification models. And it's pretty exciting, some of the results that indeed you're able to identify either one of the extremes, but mostly accurate the negative extreme with quite high accuracy. So for instance, when we're using random forest, we can capture about 90% of the materials that were predicted to be low yielding were actually low yielding. And now if you're thinking about, logistics and resource allocation, if we can precisely discard materials that are not high yielding, it really improves your testing capacity, right? Because we looked at our data, we may be able to throw away 50% of lines that have no potential, but you still have the same resources. So you can increase the number of testing locations and so on for the lines that do have potential for performing. So the other thing we can do with this is we get the probability of each one of the lines to belong either to the high yielding or low yielding. And with that, we can make decisions with a little bit more confidence. So looking at the low yielding section, we can comfortably discard anything that has high probability of being low yielding and still be able to improve testing capacity. So I don't think we are in that stage. Maybe some people may be of really capturing what is the very best one, but as long as we can discard some that are the worst, it's still pretty good. To summarize, I still really believe that good phenotypic data is one of the major drivers for prediction models. We see difference in accuracy across models, but they're not necessarily super significant. I think the quality of phenotypic data is still very important. I think genomic prediction is a tool, right? It's an exciting tool, but it's still a tool. So it got to be beneficial, or you'll be able to save money, or increasing your efficiency, or increasing your genetic gain, most things like that. Especially for the students who may be attending today, I think thinking about the logistics and not necessarily forgetting about traditional breeding is, in my opinion, very essential to be able to maximize this. So if you understand the problems in breeding, how can you use this tool to make the problems, solve the problems better? With that, we have run a little bit over time, but I appreciate it once again, the invitation, and I'll be happy to join the group and try to answer any questions. Thank you.
Thank you so much for that, Kyle. Excellent presentation. I am going to stop the slide presentation. I'm going to invite everyone back on screen here. Excellent. Kyle, if you can join us as well. There, yeah. So first of all, a giant thank you to each of you for an in-depth and really interesting presentation from each. I also want to make a very big thank you to the people who have joined us. I don't know if while you were speaking, you were able to see the chat on the side has been very, very active. There's no surprise when you bring together a group of probably predominantly scientists. There is much helping of each other, even in the chat, which is a lovely thing to see. Also, lots of questions. Technically, we are supposed to be done in 2 minutes. Given that there is a lot of interest in this topic, are each of you okay if we were to extend by 10, 15 minutes in order to be able to capture some questions?
Yeah, sure.
Fantastic. Okay, excellent. I have received a number of questions in advance of this webinar. And then obviously there's quite a number of questions in the chat. We will try to get to as many as we can. I know that Enid already provided the QR code, and I think all three of you provided email addresses. We can provide that to in the on-demand video, obviously, and also on our website. So that, are you okay if we don't touch on certain questions, people reach out to you individually?
Yes.
Fantastic. Okay, so let's see what we can get through. I will throw questions at maybe specific individuals, and if you want to suggest that somebody else could answer it differently or better, or if you'd like to add to an answer, please just jump in. So let me start at the beginning, perhaps. Kyle, how about if I throw this one to you? If we want to get started with genomic selection, how do we know whether our current training population and phenotypic data are good enough?
Well, I think you first need to define what is good enough for you, right? I think people may have different thresholds of what is good enough. If you have a really tough phenotype to collect, your good enough may be different than if you have a really easy to collect phenotype. So I think it all depends a lot in what are you trying to do. For us, to give a little bit of perspective, we didn't have much genotypic data to work with. So we try to do, we have the resource to do it. So we just decided to go ahead and genotype everything that we could, that we had good phenotypic data to use. And I guess in that sense, there's a lot of different things, and Diego can touch base here. You can play with your data with different cross-validation scenarios, and so you can, in simple terms, mix everything around to see how well can you predict things. And I think I would start there, but I think a lot depends on what you need to do.
Yeah, in our case, we decided good enough based on the on the heritability. So we know that, I mean, I'm no breeder, I'm learning from you guys breeders, but based on that, we can have an idea of how good the data that we have at hand is mimicking the reality. If heritability is low, and we are getting very close to the threshold of heritability, I mean, it should be fine. Heritability is high and the results are very low, I mean, it's not worth it. So again, it's gonna vary, it depends on the trait, crop, environment, etc. But it's kind of an idea how we can evaluate that.
Ina, did you want to add to that, or? Oh, you're muted there.
I'm sorry. I agree with what they have said, and I agree with Diego about heritability, and that's one of the most important. And also highlight that there is not a model that fits all. Like depending on the crop you are focusing, the specific traits that you manage. That's very important to take into account as well.
Okay. A question from Pedro says, do you believe someday we will use genomic selection to select the best genotype, or will we always use it to discard the worst one? I see smiles from Enid and from Kyle. Diego, how about I throw it to you?
I mean, it's a great question. And by definition, genomic selection is kind of a concept to be able to discard or to screen a large number of genotypes. So it might be the case, and that's something that we always discuss with the students, that we don't care that much about how well you are predicting the genotypes, especially those are in the middle of the distribution. So we care if we're able to identify those in the top, right? Because we want to advance those. Or alternatively, we are able to identify the bad ones, might be another way to increase the testing capacity. However, I think the goal for us is to modify the models, to change the concepts, to be able to identify that reduced number of percentage of genotypes that we want to advance. So obviously that is going to require to work again with the models, so not with the current methods that we have now.
A question from Anand. Which prediction model is best for the GBB's prediction?
That's a good question, and it depends on the data that we have available, because if we have a lot of different data types, we might get confused with the model. So we are start stealing signal. The cornerstone that we are using now is something that has been used in the last 3 years, something that we call GBlock model. It's based on covariance structures. It's very easy to implement. Computationally speaking, it's very convenient. And if we want to improve predictability, that is different than having a very good estimation of the GGBs, we might use interaction models. But again, it is going to vary what is the stage of the breeding program. What do you want to predict, or what do you want to estimate?
Excellent. A question from Ignacio. He says, question to all, it will depend on crops, but how many steps should be good to make a proper genomic selection? Ina, do you want to start out?
That's a very good question. And I would say, I don't know if Diego and Kyle will agree with me, but again, depends on the crop and depends on the coverage of the genome. So I don't think there is a magic number for all the crops, but if you cover the whole genome, you have a good coverage of your genome, it could be good enough. Or depending on what you are happy with, right? But in the case of SNPs, you need to have a good coverage.
Okay, and Diego, you looked like you wanted to add to that.
Oh no, just I wanted to add a little bit. Yeah, it's interesting because some time ago we were running some analysis with a chickpea population that has like 14 million markers, and we got similar results using 2000. So this is not, we're not trying to find that set of major genes. So as long as we're able to have a very good covariance structure, it won't matter that much. I have seen cases where we use 500 markers and the results are more than good enough.
I would also add to what Diego mentioned that, we run different scenarios. Obviously, I guess most of our answers are going to be it depends, because it really depends on a lot of different things. But depending on what kind of approach you're taking and what kind of model you're using, right? If you're not necessarily using things that are going to shrink some of your predictors to zero or something like that, you can actually see a bit of decrease in your accuracy by overfitting too many SNPs. So I think obviously it's trait dependent, it's crop dependent, but it's one of those that there's not necessarily a magic number or a rule of thumb. I guess what Enid said is very good. As long as you have good coverage for that specific trait you're working with, you should be okay.
Okay. Arturo has another numbers related question. It says, question for Kyle. For those 250 to 300 advanced lines you select with genomic selection, how many individuals do you select per population? Would you be comfortable selecting 300 individuals from 300 different populations if the predictive yield was the best for those 300 individuals?
That's a good hypothetical question. I think it's very unlikely you're going to see something like 300 from 300 different populations. But following the question, I think, and this is something we may not have touched base enough today, that with genomic prediction, depending on your training set or how you're doing things, you may start narrowing down your genetics too much in one way. And breeding it's an open loop thing. If we think we can get things done just recombining the same materials, you're going to reach a dead end, not too far from today. So I think following on his questions, one of the things that we also need to think about when making selections based on this is the diversity of the materials. So yes, if you get 300 pedigree and you get the 300 best lines from each one of them, that will be perfect. It will be a beautiful scenario. Doesn't work like that. You're going to probably have a handful of populations that are going to yield the best materials, but you got to balance genetic diversity also, because it runs away pretty quick.
Excellent. Okay. A question from Carolina. What tool do you use to predict genetic variance and progeny means to select promising crosses?
David, do you want to go first?
Okay, so to predict variances and predict the mean of the crosses. Yeah, I think we can use, again, it's not like reinventing the wheel. We can use conventional G-block models where we can have associated to each one of the different values that we're predicting, besides the mean, the standard deviation. And it's interesting, many times when I'm teaching to the students, I'm telling them that we might be sometimes biased to think that the most important parameter of a distribution is the mean, but variance is also important. So that's a very good question.
Just to add a little bit to what Diego said. So the basis of what we did is it's a well-known publication that's called PopVar, which is our package developed in Minnesota. So basically what we do, we know the recombination between two parents, so you can simulate hundreds of thousands of progenies, whatever computational power you have. So for each one of the parents, the combination of parents, you can have whatever number of progenies you simulated based on recombination. And then with that, you can start predicting how each one of those individuals are going to perform, and then you take care of variance from that as well.
Excellent. I have a question here that is not about the data set, not about the specifics. It is about the dollars and cents. So implementing GS requires investment in genotyping and computing. What kind of return on investment can a breeding program realistically expect? The $1 million question, Sam.
Yeah.
Enid, can I throw that to you?
Yeah, for sure. I can start and they can follow. I think you cannot predict the return on investment, actually. You start working, you have the original investment, and again, it's how you play your cards, right? You can get a wonderful variety, and then you can market it in the wrong location or in the wrong market, and then you don't have the right return on investment. So it's very complicated to predict a return on investment, but the last thing you can do is like not do things because you are scared of a bad return. But and also like programs work with these tools already and they get return. They get good varieties. Caio program is one of the examples, and many of our clients as well. So this works. There are practical examples of it, and the only thing you can do is play your cards, I guess.
Okay, everyone. Though I hate to do it, I think that we should wrap up our questions. Each of our contributors have generously offered to answer additional questions via email, if you'd like to follow up with them directly. Enormous thanks goes to Enid, Diego, and Caio for so generously sharing their extensive knowledge. This was a very in-depth discussion, and I hope that our audience got as much out of it as I did. Once again, we will be sharing this live webinar via an on-demand version, and we will also have it available on our website. So it will be sent to everybody who signed up for the webinar via email, and as I say, also available on our website. Many thanks for joining us this morning. We will see you again for our next innovation webinar. Okay, hope that works.